Hi, my name is Katharina Waury. I'm a PhD candidate at the Vrije Universiteit in Amsterdam, and today I will present our work on sequence-based prediction of extracellular vesicle-associated proteins. Extracellular vesicles, or short EVs, are membranous structures that are secreted by virtually all cell types. Based on the biogenesis and their size, you can differentiate three major subtypes, which are exosomes, microvesicles, and apoptotic bodies. They can contain many different types of molecules, such as nucleotides or metabolites, but here we were only interested in protein cargo. Moreover, we only looked at exosomes and microvesicles, as those are thought to be involved in cell-to-cell communication and further have also been implicated in pathologies such as cancer or neurodegenerative disease. EVs are a good biomarker source as their protein cargo indicates what is happening in the donor cell and also because they're easily accessible from body fluids. We were therefore interested in what affects protein-EV association. While being a good biomarker source, EV-associated biomarkers require specific detection protocols that involve the breaking up of the vesicle to actually detect the cargo. Therefore, another point we were interested in is it possible to predict EV association, especially if only the protein sequence is known. At the start of our workflow, we created an EV protein and a human protein dataset. The EV proteins we took from databases Physicopedia and Exacorder, and the human proteome dataset we took from UniProt. For this human proteome dataset, we generated the features we thought might be important for EV association classification, such as amino acid composition, physicochemical properties, and secondary structure and surface accessibility prediction. We also use created annotations, mainly post-translational modifications. We then filtered our datasets by removing low-count proteins from the EV protein dataset and removing non-MS detectable proteins from our human proteome dataset. We included this step because early on we realized that EV proteins showed a higher molecular weight distribution compared to non-EV proteins. We then became aware that detection by mass spectrometry, which is the dominant detection method for EV studies, is biased towards higher molecular weight proteins and could therefore also bias our analysis. In the plot, you can see that after removing the non-MS detectable proteins, the distribution of EV and non-EV proteins is much more similar to each other. After filtering, we created our datasets. We designated all proteins that were still in the EV protein dataset as positive, so EV proteins, and all human proteins that were not included in the EV dataset as negative, so non-EV proteins. We did this for general EVs, for exosomes, and for microvesicles specifically. For all three of these datasets, we created random forest classifiers, one using all features that we generated and one using only the sequence-based features, leading to 6 models in total. And those models we then analyzed to see which features were most important for the correct classification. We then looked at the performance of our 6 classifiers to answer the question if EV association prediction is a possible task. As you can see, for general EV prediction, we received an area under the curve of 0.82 and an AUC of 0.75 if we only use the sequence-based features instead of all of them. A similar trend you can also see for the exosome and microvesicle specific predictor, where in general the performance was a little bit better, indicating that this is an easier prediction task. To understand why EV association prediction is possible, we then looked at the features that we generated to see which ones were most important for classification and to see which features showed the biggest differences between the EV and the non-EV group. A few examples of physicochemical features that we generated with significant differences between the two groups you can see here. Those indicate that EV-associated proteins are in general larger, more stable, with a lower isoelectric point, so more acidic, and with lower hydrophobicity compared to non-EV associated proteins. One extremely important type of feature that came up during our analysis is post-translational modifications. As you can see in the bar plots here that show the fraction of annotated proteins, both in the positive and negative group for EVs, exosomes, and microvesicle datasets, you can see that in the vesicle-associated protein group, you always find more of the PTM types, and also those were really important features for classification. Especially for palmatolation, which is a type of lipidation, you can see a clear enrichment in the EV group, and also was for all 3 of our classifiers the most important feature. And now to summarize our most important findings. First off, while EV databases are a valuable resource, they are obviously biased towards proteins that are detectable by mass spectrometry, and therefore towards proteins with a higher molecular weight. It is therefore necessary to correct proteomics data that comes from mass spectrometry. We also showed that the prediction of EV association is a possible but not trivial task. It is possible since we achieved quite decent AUC scores with a simple random forest classifier, but it is not trivial since we also needed a wide range of features to achieve that kind of accuracy. We showed that there's clear differences in the physicochemical properties of EV proteins compared to non-EV proteins, showing that the sorting of proteins into vesicles is not random. Especially PTMs seem to be strongly involved in the sorting process since they are that strongly enriched in the EV group, and they should be examined further to understand the function. Thank you for listening, and I want to acknowledge our group at the VU Amsterdam, especially my colleagues Der Gogishvili and Sana Aben, who I worked with on this project, and the Myriade Consortium.